You are probably wondering why I have not set that typical caboodle tasks that you hopefully are now nice and used to. So the reason for that is we are now implementing, as a whole school, we're implementing a new structure for your home learning. To do this, and why we're doing this, is because we want to ensure that you are getting the best deal possible, and we're going to try as best as we can to now move to a form of home learning which is much more replicating a classroom-based scenario. So you will have already noticed from the first slide the PowerPoint templates that I'm using are exactly the same that you'll see in class, and I know that you have all desperately missed my challenge grids, so I couldn't wait to get you to do one of them. So the way this is going to work very generally is I'm going to set these PowerPoints on Show My Homework every week, and the plan is that throughout these PowerPoints you have a few mini tasks to complete. The first mini task is, as you can see on the first slide there, is a challenge grid. So the way I've done it is the next slide is a blank challenge grid where you can put your answers, and then the slide after that is the answers for the challenge grid which you can self-mark. So the mini tasks that you see within these PowerPoints are mini tasks for you to complete and for you to mark yourself. What will also accompany this on Show My Homework is a final piece of work which I expect you to submit to Show My Homework. That will be external to the PowerPoint, but it will still be related to it. So as you can see, we're recapping reproduction because it was one of the later units we did before schools closed. So we're going to recap reproduction over the next couple of weeks, and there will be a single piece of work that I need you to complete and submit to Show My Homework, which I'll provide feedback on. As always, you are welcome to make some supporting revision notes, and as always you are welcome to submit them to me. Many of you have been doing this already and it's been great to see. And if there are any sort of major issues or concerns or anything that I want to address in those notes, then we can do that on a one-to-one basis. So your plan now is just to go through this PowerPoint slide by slide. I'm going to have sound bites like this on most of the slides, flick through them, listen to my boring voice, and hopefully it's going to provide a bit more structure and give you a better deal in terms of your home learning. Hello ladies and gents. Okay, so before I wanted to actually dive into the reproduction unit and the lesson itself, I just want to quickly recap some things that we have already been taught from previous topics. So there's four main things that we just need to quickly revisit and ensure that we have that sort of foundation knowledge before we can move on. So these four things were found back in Year 9, and they were actually found very early in Year 9, so it's understandable if your memory of that is a little bit hazy. So first of all, the cell cycle. So every cell, if we think of it as a life of a cell, goes through some key stages. So first of all, we have stage G1. Just to be clear, you're not expected to remember the individual stages exactly. You're never going to be asked a question in your GCSEs about each individual stage, but it just helps to lead into what we're going into today. So G1 is just the cell going about its normal life. It's undergoing certain functions that it's expected to do. It's essentially just doing its job. Lovely stuff. Then we move into phase S, or S phase, and this is where we see the duplication of each chromosome. So each of the 46 chromosomes are replicated. All the DNA is replicated. And this is where we see that typical image. So when you think of a chromosome, you think of that X shape. Well, that is actually two duplicated chromosomes. So what you're looking at there is technically two chromatids or a duplicated chromosome. So S phase is all about the replication of the DNA, which in part is replication of the chromosomes. So then we move into stage G2. And this is essentially the final checkpoint for the cell. So this is where the cell is double checking all of the replication. So you can think of it as one person has the job to replicate all of the DNA, and then that moves to another person to make sure that every piece of DNA has been replicated correctly. This is very important when we think about cancer, because we know that cancer forms from typically a failure of this checking system, where cells are allowed to get through this checkpoint without being checked properly. So the cell cycle moves from G2, where it's getting double checked, and if the DNA is replicated correctly and it's all looking good and there's going to be no mutations or no major mutations, then we can move into mitosis, which is cell division. So that leads me into that second point, mitosis, which is cellular division. It takes place in most of our cells. Mitosis will produce identical daughter cells. So if you have the one cell as the parent, the two cells that are produced from that parent will be identical. They are clones. This is the main use for this is tissue repair. So if you cut your skin, you're going to need some more skin to replace the skin that you've just cut. That's going to be mitosis that is going to reproduce that skin cells. And as well as that, growth. So for you to get from, just a small baby to get to the height you are now, you needed your cells to replicate over and over again to produce enough cells for you to be the size you are now. As well as that, we have specialised cells and embryonic stem cells. So just to link them two together, because they're closely linked, a stem cell is an undifferentiated cell. Specifically, an embryonic stem cell is how we all started. So an embryo is when you are in fetus, so before you are born, the very, very early stages of life, you are an embryo, and as those cells are dividing, they are called stem cells, and they are reproducing at a massive rate. And we go from a stem cell, which is undifferentiated, so it doesn't have a specific role yet, but it has the ability to turn into any cell. We then go under a process called differentiation, which then produces our specialised cells. So we go from an embryonic stem cell to, for example, a sperm cell, or from a stem cell to a red blood cell, or to a stem cell to a neuron, a nerve cell. So we all started as a stem cell, but you notice that those three cells I gave examples of there, they're all very different. So these four things are sort of essential to understand before we can move into the next phase of this unit. Okay, so the first thing that I want to recap and just go over, funnily enough, in the reproduction unit, are the two types of reproduction. So in nature, in biology, we have been able to categorise all the types of reproduction from all the organisms and all the different animals and plants on the planet, categorise them into two types of reproduction. We have sexual and we have asexual. So when you think of reproduction and what that is and what that means, what we typically think of is sexual reproduction. And what that means in biological terms is a fusion of a male and a female gamete. So we should know that gametes are the scientific word for sex cells. So in humans, that would be a sperm and an egg cell. So sperm being male, egg being female. And for a plant, for example, you have the female sex cell, which is the ovum, and the male sex cell, which is pollen. So one of the key features of sexual reproduction is that it produces a mixing of genetic information, some from mum and some from dad. And the good thing about this is that it causes variation. So variation is very good because it increases the longevity of a species. So what that means is, for a key example, is that if we have variation in the genetics of each person, then a disease is much less likely to wipe out the whole population. So we could apply this to coronavirus at the moment. So we know that coronavirus is affecting different people differently. Some people are having severe reactions to the coronavirus, and some people not so much. Some people get the disease and get the infection and are completely unaware that they even have it. That's what we call asymptomatic, and they show no symptoms whatsoever. And the reason for that is variation. So if all of the humans on the planet had the same genetic information, if COVID-19, that specific virus, was able to kill one person, then it would most likely spread and kill everyone. But because we have variation, it affects some people worse than others. As well as that, with sexual reproduction, and I'll go into this on the next couple of slides in a bit more detail, but with sexual reproduction, we have the formation of gametes by a different type of cell division called meiosis. So the cell division that occurs in most of our body cells is mitosis. However, when you want to produce a sperm cell or an egg cell, that process is called meiosis, and I'm going to go into that in a little bit more detail in a second. So sexual reproduction, it's all about the fusion of a male and a female gamete, and it includes the mixing of genetic information, which is always good because it means that we have variety and variation in the species. So asexual reproduction. So whenever you typically put an A in front of a word in English, that means the opposite of the following word. So if sexual reproduction is the fusion of male and female gametes, then asexual reproduction does not include that. So asexual reproduction only comes from one parent. So there's only one parent needed for asexual reproduction to take place. Now, because of that, there's no mixing of genetic information, and it means that the daughter cells from the cell division, they are identical. So with asexual reproduction, you are producing clones of each other. Now, there's a couple of advantages to this. It's much quicker. So this is why we see asexual reproduction typically in microorganisms. So bacteria, for example, the reason why it is able to grow so rapidly is because it typically undergoes asexual reproduction. It's also much easier because it only requires one parent. So we know in the biological world with the more complex organisms that require sexual reproduction, they require two parents, and that often involves finding a mate. So it's easier to undergo asexual reproduction because you don't have to have that partner involved. And as well as that, because you are producing clones, asexual reproduction, mitosis is involved. So there is no such thing as meiosis when we're talking about asexual reproduction. Meiosis only applies to sexual reproduction. So thinking a little bit more about sexual reproduction and staying on that sort of trend there, one thing that we do need to understand in a bit more detail is meiosis. What exactly is meiosis and how is it different to mitosis? Annoyingly, and you do get this quite often in biology, they are very similar sounding words, but they are very, very different processes. So yes, they are both forms of cell division. However, what is produced is very different. So very generally, meiosis leads to non-identical cells being formed, which means they are not clones. With mitosis, we get those identical cells, they are clones that are formed. Now, meiosis only ever takes place in the reproductive organs. So in males, meiosis will only ever be occurring in the testes, where we are producing sperm cells. And again, in females, you will only ever find meiosis occurring in the ovaries, where you are producing eggs. So meiosis leads to the formation of a sperm cell or an egg cell, and it only ever occurs in the testes and ovaries. So meiosis produces haploid cells or haploid cells. Now, haploid cells are the fancy term for sperm cells and egg cells. The key feature of these is that they only have 23 chromosomes. So they have half the number of chromosomes that you have in all of your other cells. Now that's important because when the sperm cell, which has half, and the egg cell, which has half, when they meet, to form you then have a full set of chromosomes. You have that 46 or that 23 pairs. So this soundbite is linking not only to this slide, but it's also linking to the next slide. So with the next slide, I've shown you the stages of meiosis. Now let me be clear here, you do not have to remember each individual stage and what happens. But what you are expected to remember are a couple of the key features. So the first key feature, and it might help to flick back to the fourth slide here where we looked back at mitosis. So with mitosis, only one cell division occurs. You get one parent cell and it produces two cells. That's one cellular division. However, with meiosis, you have two cell division that occurs. So we have one parent cell which divides to form two cells, and then those two cells divide again to form two gametes. So you go from one parent cell to four daughter cells. Now, as well as that, in each division, so in each of those divisions, you have what we call a crossover of genetic information. So that's why in that picture, if you look at the chromosomes, you'll see that certain ones, the colour have changed. So we go from red to blue, and you can see a mixture of that. What that's showing there is the crossover of genetic material. And all that's happening there is that is ensuring that we have a variation of genetic information. So that just really does make sure that the daughter cells, and that is why the daughter cells are going to be genetically different to the parent, because you've had a mixing of genetic information there. So just to recap, with meiosis, we end up with 23 chromosomes as opposed to 46. Meiosis is all about the production of a sperm cell and an egg cell, so it's only going to occur in the testes or ovaries. We have two divisions that are occurring as opposed to one in mitosis, and we have a crossover of genetic information which gives us that variation that we want. Now, the final section that we're going to look at in terms of looking at sexual reproduction is arguably the most important part, is fertilization. So remember, the definition of sexual reproduction is that fusion of the male and the female gametes. And what fertilization is that precise moment. So when the sperm meets the egg, or when the male gamete meets the female gamete. So in humans, we're going to use this concept. I'm going to use the example in relating to humans, but this does apply to all sexual reproduction. So you could apply this to plants in terms of the pollen and the ovum. However, I'm going to stick with the example of humans as it's the most relevant and easiest to relate to. So essentially, with fertilization, we have three key points. So the first point is fertilization itself. It is when the sperm is meeting the egg. And when the sperm meets the egg, not only are you getting 23 chromosomes from the sperm, 23 from the egg to form a 46 chromosome cell, but what happens is we go from those two cells and they actually form a unique cell, and we actually call that a zygote. So the first cell that is formed from fertilization is called the zygote. And then from that cell will divide by mitosis. So remember, we're now dividing into identical cells to form two identical cells from there. And then you can see from the pictures, we go from, so picture B, we've got two cells there. They're going to undergo mitosis to form four cells. Those four cells are going to divide again to give us 16, from 16 to 32, and then that final picture will actually have 64 individual cells in. And this is why I wanted to just quickly recap the idea of stem cells and cell specialization. because although it's true that I'm saying that this is mitosis, some of you may be sitting there wondering, well, how if it's mitosis and they form identical cells, how do they then form sort of our skin cells and our stomach cells and our red blood cells and our nerves? Like, if they're identical, how does that happen? So all of these cells are actually stem cells. So although they are identical, they are all identical stem cells. And they all have the ability to undergo specialization and differentiation and form specialized cells. So later in development, these cells will undergo their own form of specialization. Some of them will become muscle cells, some of them will become skin cells, nerve cells, etc., etc. So how we get from these identical zygotes into then our specialized cells is that process of cell differentiation. So here, all I'm looking for you to do, it's just one of those mini tasks again. So I've given you a number of statements on the PowerPoint slide, and if you look at your next slide, you'll see a blank table. All I want you to do is read those statements and which statement applies to sexual reproduction and which statement applies to asexual reproduction. So if you're working on the PowerPoint itself, you can just copy and paste each statement into each column of the table. Make sure that you also look at the diagrams and which diagram applies to which form of reproduction, and just put them in the table. And then again, slide 12, you can then double check your marks to make sure that you've got them correct. Now, the final part of this session is looking at DNA in the genome. So we've looked at sexual and asexual reproduction. We've looked a little bit more into sexual reproduction and looked at meiosis and what that is. And we've also looked at the process of fertilization. Now, the second part of this session is all about the DNA and the genome. So what exactly is DNA? So DNA is essentially all of our genetic material that's found in our cell, in our bodies. So all of our genetic material in our body we can call the genome, and the genome is made up of DNA. Now, DNA is a structure that looks a bit like a twisted or a spirally ladder, and we call that a double helix. So double meaning two, and helix coming from the word like spiral. So think of it as a ladder that's just been twisted and spiraled around, and we call that a double helix. So DNA is a polymer. Now, all a polymer is just a large molecule that's made up of repeating smaller molecules called monomers. So in the example of DNA, the monomer for DNA, so the repeating smaller molecule, is called a nucleotide. Now, you don't need to remember this for your exam, but just to extend yourself a little bit further, and you will look at this further if you pursue science post-16, but all a nucleotide is made up of 3 parts. So it's made up of a 5-carbon sugar, it's made up of a phosphate group, and a nitrogenous base. So if you were to Google DNA structure, you might see that some of the pictures on the sort of rungs of the ladder, you will see letters. Now, those letters are called nucleotide bases, and they are what actually code for specific proteins, which I'll go into in a bit more detail on the next slide. So we know that the genetic material found in our cells is called DNA, but we just want to go into a little bit more detail of where we found our DNA, and specifically what we look at if we were to dive into the nucleus a bit more. So we know that the nucleus of the cell is where the genetic material is found, and we know that the genetic material is DNA. We know that the nucleus contains 23 pairs of chromosomes, or 46 chromosomes, but what exactly is a chromosome? So you can see from this picture, and it is not to scale, so DNA is not that big, the molecule is not that big, but the chromosome, the picture or the diagram sort of explains the point nicely. So all of our DNA is wrapped up, really tightly bound into these structures called chromosomes. Now, within our chromosomes, we have, if we were to split them into little sections, we would find that throughout the chromosomes, and so throughout the long strand of DNA that is wrapped up in a chromosome, we have little sections that we call genes. Now, genes are very important because they produce our features. So these small sections of DNA, which we call genes, they produce our features. Now, you may recall back in primary school or even in year 7 or 8, DNA being likened to a recipe book. So DNA is the recipe book, and it has all the information required to make the cake, or you, for example. Now, each gene codes for a sequence of amino acids. So it's like putting those ingredients together and it forms an amino acid. Now, you don't necessarily need to know what an amino acid is, but you need to know that when you make enough amino acids and you put them together in a certain order, they produce a protein. Now, protein, yes, it's similar, it's the same structure as the protein we talk about in terms of our diet and getting protein, but proteins in terms of sort of human biology, all a protein is a very generalized term for a structure in the human body that has a specific function. So, for example, we could have a protein that is a digestive enzyme, so it breaks down our food, or we could have a protein that is a hormone. So it could be the hormone that triggers the start of ovulation in the menstrual cycle. Or it could be a protein, or in this example, a combination of proteins that produce a specific hair colour or a specific eye colour. So proteins, it's very hard to get your head around what a protein actually is, and that's because it's a very generalised term. But just understand that a protein is something or a structure in the body that has a very specific function. So you don't necessarily need to know how DNA is turned into proteins. That's a process called transcription. So how DNA is then turned into amino acids, which is then turned into a protein. You don't need to know the specifics of that process. That comes in sort of A level. But you do need to understand that a length of DNA is called a gene. And a gene codes for an amino acid. And a string of amino acids is called a protein. And proteins produce our features, they produce our hormones, they produce our enzymes, things like that. So, as we've already understood by now, the genome is the term used to describe all of the genetic information found within the body. So, one thing that we do need to understand for GCSE is something called the Human Genome Project. Now, it's up there with some of the greatest discoveries ever in science. When we completed the Human Genome Project, it was a massive, massive jump for human biology. So what the Human Genome Project was, it was a massive research project to map out all the genetic information within the human body. So if we go back to the analogy of the recipe book, and what we wanted to do is we knew that the DNA and the genetic information in the human body was a recipe book. We knew that it included all the information, all the ingredients to produce all of our features, all of our enzymes, but we didn't actually know the ingredients. We didn't know what gene did what, and we didn't know exactly where to find it. So the point of the Human Genome Project was to complete that recipe book. It was to get an understanding of what gene does what in the human body. Now, this started in 1990, and it actually took 13 years to complete. So now we have the whole human genome completed, and we can study the human genome, and we know what genes cause what things. So the main sort of consequence of this is found in medicine, and it helps us understand diseases so much better. So not only are we looking at genes that are linked to certain diseases, but it also helps us treat those diseases. It also allows us to get a better understanding of inherited diseases. So inherited diseases are disorders that you bring down from your parents, so you get them from your parents. And lastly, it's not discussed as much, but it is just as important, is it allowed us to trace our historic migration patterns. So there's a link for a video that nicely sort of discusses that you can look at. But it allowed us to truly understand that we as a human race, we did all originate from Central Africa, and we have then ventured out throughout time. If you did want to do a little bit further study, when I was at university, I had a lecturer called Professor Colin Smith from the University of Brighton, and he was actually the first person in the world to publish his genome, so his own genetic material, into the public domain, so you can actually access his genome. Now, what this meant is that when you analyse his genome, it allows us to not only identify certain diseases that he is most likely to get, or sorry, most likely probably isn't the right word, he has a predisposition to get. So he might be more likely to get heart disease because it runs in the family, or he might have certain genes that are heavily linked to a specific cancer. So this means that we can't exactly predict the future, but if we are aware that Professor Colin Smith has certain genes that are heavily linked to cancer, then we can at least prepare for that, and he might, for example, get more tests than the average person for that specific cancer, so that you can catch it early. As well as that, one of the key features, and this is happening right now, to be honest, we're still trying to perfect this, is something called personalised medicine. So by analysing a genome, if, for example, let's go with a very basic example of hay fever. So with hay fever, there's a few drugs that you typically buy, so you pop down to Boots and get your hay fever tablets. So you get, the two main types are loratadine and cetirizine hydrochloride. They're the two main hay fever tablets. There are a couple of others, but they're the two main popular ones. So what we could do is study my genome and understand my genes, and then from that we could identify which hay fever tablet works best for me. So instead of me spending years to figure out that cetirizine hydrochloride is actually the best one for me, you could simply understand my genome, analyse my genome, and we can figure that out. Now, I understand hay fever probably isn't, the biggest and best example for that, but this can have massive implications when we're talking about treating heart disease or treating cancers. If we can identify which treatment is best, instead of sort of trial and error, trying one drug, oh, that doesn't work well, okay, let's try this one. If we can analyse a genome and make that decision before we've even trialled it, that means we're getting the best treatment for the patient from day one, as opposed to maybe wasting time trying to find that right treatment. So it's very, very important.